All living systems emit light. When plant cells undergo oxidative processes, they produce a continuous, spontaneous glow. This is ultra-weak photon emission, or UPE. It is a direct result of the generation of reactive oxygen species. Reactive oxygen species, or ROS, are chemically active molecules containing oxygen that are produced when a plant is enduring environmental or physical stress. To capture this light, researchers use a highly sensitive charge-coupled device, or CCD, sensor. A CCD is a specialized sensor that converts low-level light into electronic signals for imaging. This image shows a healthy sunflower leaf captured by a CCD sensor inside a dark chamber. The baseline photon emission here is low. Physical damage changes that light output. When a leaf is pierced to mimic an herbivory attack, a form of biotic stress, the sensor detects an intense burst of blue and green light clustering around the wounds. Severe environmental factors, like drought, trigger a different reaction. This panel shows a leaf suffering from water deprivation, an abiotic stress, which produces a high-intensity red and yellow heat map spreading across the entire leaf structure. Comparing these states side by side reveals the scale of the stress response. The healthy control leaf emits a maximum of 38 counts per second. The drought-stressed leaf reaches 1,617 counts per second. Measuring UPE provides a real-time quantification of stress, but its diagnostic application is found in the kinetic dynamics of how this light fades over time. In modern precision agriculture, acquiring data quickly is the primary operational bottleneck. Mapping the full time course of a crop's stress response requires continuous monitoring as the light fades. For large-scale sunflower production, prolonged observation of individual leaves is logistically difficult. Agronomists need to understand the decay kinetics, the specific rate at which the UPE signal drops after the initial burst of reactive oxygen species. This led researchers to a specific mathematical question. Could the initial intensity of the light alone predict the plant's physiological trajectory over the short term? Solving this would allow for a diagnosis using a single instantaneous photon snapshot, reducing the time required for continuous recording. To test this, researchers tracked photon emission of both healthy and mechanically pierced plants over 5 consecutive 60-second intervals. This chart tracks the time series data. Green lines for pierced plants and black lines for healthy plants both show a sharp drop between the first and second minutes. Zooming in on the healthy control group reveals a predictable exponential decay curve. This curve maps to the plant's biology. A localized burst of reactive oxygen species is neutralized by the plant's surrounding antioxidant defenses. An exponential decay curve is defined by 2 variables, the initial biophoton intensity, denoted as I0, and the dynamic slope of the decay, known as m. Because biotic stress follows this exponential decay, its trajectory is governed by mathematical rules. This predictability is the requirement for a forecasting algorithm. The challenge is calculating the entire decay curve when you only possess the starting data point. The research team hypothesized that the initial intensity of the light and the steepness of the exponential decay slope are coupled variables. Looking at this comparison, we can see how the height of the first measurement bar relates to the dotted trend line. A higher initial reading pairs with a steeper drop-off. To prove this, researchers plotted the data from the biotic and control samples onto a scatter plot, mapping the initial intensity against the decay slope. A logarithmic trend line through these points yields an R-squared correlation value of 0.73, confirming a statistical relationship between the 2 metrics. The graph shows that higher initial photon counts correlate to a steeper decay slope. This suggests that the decay rate is a physiological property identifiable from the first pulse of light the plant emits. The next step was translating the scatter plot into a functional equation for use in the field. The core model for the decay is y of n equals I0 times e to the power of m times n minus 1. Below that is the logarithmic parameterization derived from the scatter plot, m equals 0.5298 minus 0.102 times the natural log of I0. Substituting the second equation into the first allows you to solve for the decay slope using only the initial intensity. This removes the need to physically measure the slope over multiple minutes. When an initial intensity data point is provided, the formula instantly plots a predicted exponential decay curve across the 5-step time axis. Researchers can now use this model to forecast the expected decay intensity curve from a single snapshot. This derivation addresses the speed bottleneck in precision agriculture, providing a rapid diagnostic tool to identify and quantify acute mechanical damage. Predictive models must be tested against boundary conditions, which leads to the data representing severe water stress. This chart shows biophoton emission bars over time for plants suffering from drought. Researchers applied the algorithm here to test its limits. Instead of exponential decay, these trend lines are irregular. Panel A shows an upward trending emission, while panel C forms an oscillating wave. Because the light output fluctuates instead of fading out, the calculated decay slopes for this group hover around 0. The predictive algorithm does not map these shapes. Applying an exponential equation to this non-exponential data results in systematic errors. The model underestimates the light output at time step 2 and overestimates it at steps 4 and 5. Water stress increases emission magnitude, but it also alters the underlying mathematical decay seen in localized stress. Why does systemic drought produce oscillating right signatures, while physical piercing produces a predictable curve? The nature of the threat is different. Piercing a leaf is an acute, localized injury managed by local antioxidants. Drought is a systemic crisis affecting the entire organism. To survive drought, plants rely on retrograde signaling. Retrograde signaling is a pathway where organelles, like chloroplasts, send signals back to the cell nucleus to trigger systemic survival mechanisms. These signals travel through the plant via auto-propagating ROS waves, moving between cells to alert distant tissues of the stress. The oscillating signatures captured by the CCD sensor are the visual manifestation of these ROS waves pulsing through the plant as it maintains its internal state. The kinetics of biophoton decay differentiate abiotic from biotic stress based on the specific shape of the light decay. By capturing initial UPE intensity, we can statistically predict acute mechanical stress, and by identifying where that model ceases to apply, we can diagnose complex systemic drought. It is a non-invasive diagnostic paradigm for crop management.